In this lecture, we're going to look at one of the most well-known problems in bioinformatics, which is sequence alignment. We're going to see this relation between alignment as maximizing similarity between sequences, which is actually what we're going to do, and the idea in theory of alignment as finding the homologous regions between sequences. These sequences can be sequences of proteins or sequences of DNA. We are going to look mostly at examples of DNA, just because it's simpler to illustrate, but we can extend this easily to proteins. We're going to look specifically now at the algorithm of global alignment. We're going to see local alignment on next week. And we're going to see some variants of global alignment. But the idea here is to find matches between the sequences, considering everything that we are matching correctly or mismatching, all the gaps and so on. So let's start with the difference between homology, which would be what we would like to obtain, and similarity, which is what we can measure. So we know that organisms and their molecules are being molded by evolution, and what we are looking at any given time is just a snapshot of this continuous process. So when we have sequences that share some common ancestor, we know that what all the differences they have that we are now seeing have evolved in different branches by duplication, by mutations, by deletions, and so on. So this accumulation of changes, which is driven mostly by random factors, but then selected by natural selection, because it depends on what impact these changes have on the organism. Whether if it's affected by selection, it can be greatly filtered out, and so we have only those things that worked. In some cases, there is not much selection pressure, so there can be genetic drift, simply the random accumulation of mutations, and all these historical constraints, all of this history that results in two sequences that we look at nowadays. For example, genes, the gene that has the same function in two different organisms, and that we know by the similarity that it descends from a fairly recent common ancestor. We cannot see this process that led to all these changes. We can only see the result that we have now. So what we would like to do with sequence alignment is to try to find a way to match the different elements of the sequences in order to determine, for example, if they are very homologous or not, if they have a closer relation in evolution or a more distant relation, and also to try to find the matches between the regions in one sequence that are homologous to regions in the other sequence. That is, those places descend from the same original positions in the original ancestral sequence, but then may have accumulated some differences, some mutations. There may have been some insertions, deletions, and so on. But we would like to find this match in order to map all of these evolutionary changes and have a way of seeing the homology between the sequences. So this can help us measure the divergence during evolution. It can help us identify which regions were more conserved or less conserved, and so on. Okay, so this is the scientific theory and the scientific model for the specific case of sequences. However, we have a constraint here, that is the information that we have. So ideally, we would want to find a match between these sequences and to represent the matches between different positions by adding this extra character, for example, this dash here, to represent these gaps in order to align the sequences properly. Note that these gaps are not present in the actual sequence. So this would be one sequence, this would be the other sequence. There are no gaps in these molecules, in these bits of DNA, for example. But when we try to find the matches the corresponding nucleotides in one sequence and the other, we need to have a way of doing this mapping. So we add these spacers just to make so that these two match together, and then this one does not match anything on the first sequence. This part of this sequence does not match anything on the second one, and so on. So these gaps, these dashes that we are inserting, are just a way to help us represent the matches. And ideally, we would like this alignment to express the evolutionary history of these sequences. So the result of the evolution of, for example, either a deletion here or an insertion here, and then mutations where they may differ, and so on. So this should be a representation of how the two sequences are homologous in different regions. Unfortunately, we cannot really implement this because we do not know these events. So we cannot tell where deletions occurred, where substitutions occurred, and so on, because we cannot observe this history of evolution of the sequences. So the best thing we can do is to look at the sequences as they are now and try to find some approximation that we can observe that would be close to, or as close as we can, to what we would really like, which would be to find the homology between the sequences. So we can assume that minimizing the differences between these alignments, that is, maximizing the similarities on the matching regions of the sequences, would approximate what we would get if we were looking at the mutation events and so on. So basically, we assume that there were few mutation events, so the sequences are still very similar, and if we approximate them by similarity, if we match them by similarity, we get nearly the same thing. So this is the assumption that we're going to do when we pass to the computer model and how we're going to actually align the sequences. We are assuming that the best alignment corresponds to the best match between these sequences, the greatest similarity between the sequences, as an approximation for what we would really like to know, which is how they evolved and what kind of changes they accumulated over time. Okay, so this is the computational algorithm. We need a way to score the alignment, to check how good the alignment is, and we're going to try different alignments, different combinations of matching the parts of the sequence, and we're going to find the best one. So the one with the most matches between equal nucleotides, for example, and the smaller number of gaps, and so on. So the first thing we need is a way to score our alignments. This is a very simplified scoring scheme. We're going to next week look at these scorings in a bit more detail, but for now we're going to use just this simplified set of rules. If we have to put a gap somewhere, then we count a penalty of 2 for each gap we insert. If we have an identity, that is, if a character matches with the same character on the other sequence, then we add 1, and if we have a difference, we subtract 1. So here we have a -2, the minus sign there disappeared, another -2 here for this gap, a +1 for this identity, +1 here, and so on. So we can score the complete alignment, and we can compute that the score for this particular alignment is 1 positive. Different alignments would have different scores, and the idea now is that we're going to try different alignments, different combinations of these matches, and try to find the best one. One problem here is that there are too many possible alignments. So if you can imagine, what we are doing here is that we can align this one with any of these characters, or even include gaps here, and then we could align this one with any character that is ahead of the one that is aligned there, and all of these combinations. So we are multiplying all these combinations, and basically if we have, for example, one about 100 residues, we already reach the number of protons in the visible universe as the number of combinations, the number of possible alignments we have. So clearly, we cannot just do this exhaustively, checking all possible alignments to find the best one. And this is the kind of problem that we can solve a lot more efficiently with dynamic programming. So remember from last lecture, we are going to break this down into sub-problems. This problem has this optimal substructure. That is, if we find the optimum for the sub-problems, then we can use that to build on the next problem and so forth. And we're going to find a good ordering for these problems so that we only need a table with the highest scores for all these possible initial matches, and then we can find the best alignment. So this is the algorithm that we're going to look at now. The idea here is that we're going to break down the problem of finding the alignment between sequence A and sequence B in sub-problems of this sort. If we consider the stretch to position I in sequence A, so this AI, and the stretch from position B for sequence B until this position BJ here, then there are three possibilities. The best alignment of these fragments will be either aligning everything and up to AJ and everything up to BJ, AI and BJ on these stretches, and then matching these two, AI and BJ, will be aligned one with the other. So the best alignments on this left part, and then matching these two. Or we could find the best alignment between everything on A up to AI, including this one, align this part with this part left of BJ, and then leave this nucleotide or this residue unaligned. So align this BJ with a gap and pay the gap penalty, or do the same thing, leaving this one without a partner, so aligning this one with a gap. So basically the three possibilities are: we align these shaded regions, the best alignment of these shaded regions, and match these two residues, or we align one of these shaded region plus the residue with the shaded region of the other and leave this one hanging with a gap, or we do it on reverse. We leave this one hanging with a gap and we align all of these with this shaded part here. So we can do this for each combination of positions of A and B, and you can imagine that now we have a number of possibilities of decisions like this that is equal to the number of residues in sequence A multiplied by the number of residues on sequence B. So basically we reduce this to a quadratic problem where we're going to fill in this matrix of length of A times length of B. So this is the general idea of how we're going to decompose the problem. So let's look at the algorithm. This is the global alignment algorithm by Needleman and Wunsch from 1970, and this would be a general method that we could apply to sequences of amino acids or to sequences of DNA. So the initial proposal was to measure similarities in amino acid sequences, but we can apply it to any sequences. And it uses that decomposition that we saw and dynamic programming to speed up the calculation and to minimize how much we have to retain at each step in order to compute the next steps. So what we're going to do is to do this decision at each combination of a position in sequence A and a position in sequence B. We need to check if it's better to align A with B. So we have this the score coming from matching A to B, which in our case can be -1 if they are not identical, or +1 if they are identical. And this we add to the best score obtained from aligning this shaded region before AI and this shaded region before BJ. So this would be one possibility. The other possibility would be the alignment of everything up to I, but excluding bj. So we align everything here, including this ai, with this stretch here, excluding bj. So we stop at j minus 1. We have the best score of this alignment, and then we pay a penalty for the gap, because now we're leaving bj without a partner, and we must insert a gap here on this first sequence. Or we do it the other way around. So we align everything up to ai, this part here. We include everything here up to bj and bj to align with that shaded part there. So this would be the best score of aligning this part with this whole thing up to bj including. And then we have to leave ai outside without a partner, so we also pay a gap penalty. So this is what we need to decide for each combination of ai and bj, and this will tell us what is the optimum score up to ai and bj. But note that now we can use this optimum score to move one step, either by increasing ai or bj or both of them, because now we have what we need, all of these scores before, which is what we need to compute the next step. So the idea is to use a two-dimensional array for this, and we're going to begin with this, initializing this matrix by considering each sequence that we want to align. So for example, this is one sequence, this is the other. And we're going to include also a gap at the beginning, because we may want to include gaps at the beginning of the sequence. So this would be, the way we can interpret this initialization is, if we just match these two gaps together, we can actually ignore them, because we are just starting from the beginning of the sequence, and this would not have any score. But if we go one one position here to the right, what we are saying is that we are leaving a gap at the beginning of this sequence and jumping the first element of this sequence on top. So this would align this element here with the gap, and we would need to pay the gap penalty. And the same thing if we keep sliding, so we keep ignoring elements at the beginning of this top sequence, so we have to add these gap penalties. The same thing for those here in the vertical. If we ignore elements at the beginning of the sequence, that is, we align this A with a gap and this G with another gap and so on, then we have to pay these gap penalties, which we stipulated would be a penalty of 2. So this basically is how we initialize the array. We consider a matrix that has the number of columns is 1 plus the length of one of the sequences. The number of rows is 1 plus the length of the number of the elements in the other sequence. And we just fill it in 0 and then the gap penalty increasing on the first row and the first column. This to represent the penalty we need to pay if we start the alignment by sliding the beginning of one sequence relative to the other. And now we can do this, because we can do this for this cell, because now we have these 3 scores already previously computed. We have the fi minus 1, g minus 1, which is this 0 here, which would be, if we are considering the potential match between this A and this A, then the previous score is simply nothing, because it's this is right at the beginning of the sequences, so it's that 0 there. And to that score, we're going to add the match between this residue and this one. So in this case, they are identical. We add 1, and this would be what we would get if we would simply consider everything that comes before, which in this case is 0, and then add 1. The other possibility would be to consider everything to position I on one of the sequences, but leave us at position j minus 1 on the other sequence. So we would come from here, because we would have to have a gap so that we would not have considered this residue here before. and then we need another gap, because now we're going to jump to leave one element out on this sequence too. So this would be the equivalent of putting one gap to match this A and another gap to match this A. So it would be going this way and this way, and we have -2 and then -2 again, it's -4. And this would be the same thing but in reverse, going from to here and then here, and that's -4 too. So now we take the best score of these three. And the best score of these three would be 1. And now we can do this for the next cell. Basically, we can do this for any cell for which we already have the 3 previous neighbors filled in. Because these 3 neighbors correspond, the one on the diagonal to the first branch here, and the other 2 to the other 2 branches here. So we have all these i-1, j-1, or fi j-1, or fi -1j. So these are the best scores from the previous possible alignment. So now we keep doing this, and we repeat this for the whole matrix. And this is the typical example of dynamic programming, because since we follow the right ordering of the subproblems, we always have the information that we need to compute the best optimal solution. We have the optimal solutions for the previous alignments, and then we compute the next optimal solution by considering the 3 possible branches and picking the one with the highest value, so with the largest value. Okay, so now we have this matrix fully computed, and we can immediately tell the score of the global alignment. So the score of the global alignment is given by this last value here. For example, the best score for aligning these 2 sequences would be -1 using the scoring rules that we decided on. -2 for the gap, 1 when they are identical, -1 when they're different. So after solving everything, note that each of the values here give us the best alignment score for each subproblem. For example, this -1 is the best alignment score for AA with A, because we have one bonus of 1 since we can align one A with the other A, but then we have a -2 penalty because one of the A's will be left out. So this -1 here is the best score for these 2 subsequences. The same, this -2 here would be for these 2 subsequences, the -2 here would be for the subsequence of A with nothing, and so on. So you can see that each cell of this matrix has the optimum alignment score when we consider all the parts of the sequence that are from the cell upwards and from the cell to the left. So when we reach the end, we have the optimum score considering the complete sequences, all of the sequence here on the left and all of the sequence on top. Okay, so now we don't want only the score, we want the actual alignment. We want to be able to write down which residues, which elements of each sequence correspond to the elements of the other sequence. So what we can do here is we're going to trace back from the last cell by going backwards on all the decisions that we took here. So if you were implementing this on the computer, the best way would be to store each of these little steps. For example, here we take a step on the diagonal because this is the best one. And so we, apart from the matrix, we also store that we took a step on this diagonal here, so that then we could go back the same way. So this would be the idea here. We're going to trace back from the last cell. Since we are not computers, I don't want to keep track of this in a different matrix, but we can just calculate for ourselves where we could have come from. In this case, we could not have come from here because we would have to pay a gap penalty of -2 to go down from this cell to this one, and the same to go right from this cell to this one. So every time you go on a vertical or horizontal, you need to pay a gap penalty, because this means you are advancing on one of the sequences and not on the other. This requires leaving a gap so that you don't advance on the other. When you come on the diagonal, you are advancing on the two sequences, so this means you are matching the two characters together, the two symbols together. So in this case, if we go from -2 to -1, it's a +1 bonus, which corresponds to this C matching a C. So we know that here we must have come from the diagonal matching the two C's. And this is what we can put here on the end. The two C's matches. Now we are here. We could have come, for example, from the side here, because it goes from 0 to -2. And this means that we would be advancing on the sequence on top. We will be using this A, but we are not using anything from the sequence on the right. So we leave a gap here because we are still on the G. We still did not use this G here. And now we'll keep tracing back. We know that this could be a diagonal from 1 to 0 by matching the G with the A. We pay a penalty of -1. So now we use both of them and we jump from this cell to that one. And now we can go from 1 to 0 matching the two A's. So this would be one possible alignment, and it would give us a score of -1. We have +1 here, +1 here, -2 here, and then -1 on this mismatch here. So this is one of the best alignments possible. It's not the only one possible, because as you can see, we have here two possibilities. We could have come from the left here and go from 0 to -2 paying a gap penalty, but we could also have come on the diagonal and going from -1 to -2 by matching this A with this G. So there is an alternative alignment here, which is this one, that has exactly the same score. And this, by the way, is intuitive because we are aligning G with A and one A with a gap, or we're doing the same thing here, but we just move the G from one place to the other, but the score will be the same. So you can find places in when you're filling the matrix when two branches have the same score, and this will give you alternative alignments when you do the backtrace to find the actual alignment. So if you're only interested in one of the alignments, it doesn't matter which one you choose because the score will be the same. If you want to enumerate all possibilities, then you need to check for each of these possible branches and do the different backtraces. Okay, so basically what we need is this matrix where we have as many columns as the length of one sequence plus 1. We need that gap at the beginning. And as many rows as the length of the other sequence plus 1. We're going to start from 0 on the corner because this is before aligning anything. And then we're going to fill the columns and the rows with the gap penalty increasing because this represents starting one sequence misaligned with the beginning of the other and padding with gaps. So we need to pay the gap penalty to do that. And then we can do this procedure of filling the matrix from left to right and top to bottom because we know the best scores, the scores for the optimal alignments for the previous subproblems. And the previous subproblems are the parts of the sequence corresponding to everything in our current cell up and everything to the left. So those are the ones that we already solved, and we have the best score for those subproblems. So we fill in the matrix in this way, and now we trace back from the last cell. The last cell gives us the score for the global alignment between the two sequences, and we trace back by checking which steps we did. When we implement this on the computer, we keep track when we are solving, computing the value of each cell, we keep track of what was the step we gave at each point. But if we are doing this by hand, we can just check which possible steps we could have taken to reach that cell. So if we are going on the diagonal, we are matching the two residues, which means that when tracing back, we need to match the two residues in the sequence alignment. If we are going left or going up, it means that we are moving along one of the sequence. That is, we are taking one residue from one of the sequence, but we are not using the other. So we leave that one with a gap. Basically, if we go up, then we need to use one of the elements in this left sequence without using any on the top sequence, because we remain on the same column. If we go left, then it's the reverse. We need to use one of the elements in the top sequence without using anything on the left sequence. If we have a tie between different branches here, then we will have multiple alignments that have the same score. This can happen, and if you check all of these branches, you can enumerate all the different alignments that have the optimal score. Okay, so let's see some simple variants here that we can apply to other situations. In this example, I am assuming that we want to align as best as possible the complete sequences. There are situations where we don't want to do that. For example, we may have a piece of genomic DNA, for example, this complementary DNA taken from messenger RNA or something like that, and we want to find where it matches inside a very large genomic DNA. In that case, we don't want to apply gap penalties to the extremities of this small piece, because what we really want to know is where it matches, and we know that there will be lots of gaps at the beginning and the end, but we need to ignore that inside this large stretch of DNA. Or we have, for example, a problem of sequence assembly where we want to match overlapping extremities, the end of one sequence with the beginning of the other, something like that. So in these cases, it may make sense to not penalize gaps at one end, or not penalize gaps at both ends of one sequence, or something like that. And this is easy to do with the global alignment algorithm. The only thing we need to do is first ignore the gap penalties on one of the rows or one of the columns. So if we ignore the gap penalties here, we are saying that we can ignore how many of the residues in this top sequence are left out of the alignment. That is, they are matched with gaps, and we don't penalize that. So we can start to align this A either on the beginning or on this one or on this one. It doesn't matter. What we want is to find out where it aligns best. So if you put this first row at 0, this means that you are ignoring the elements of this top sequence that at the beginning will be left without a match. In other words, you are ignoring penalties by padding the beginning of the alignment of this sequence with gaps, for example, like this. This we can do here without penalty, because we can just jump this A, leave this A without a partner, and this has a 0 penalty in our score. So this is the way you can use the global alignment algorithm to ignore gaps at the beginning of the sequence on the left, which is the same as ignoring leaving elements of the sequence on top without a partner. If you want to ignore gaps at the end, so in this case, let's consider we don't want penalties for adding gaps here at the end of the top sequence, or this is the same thing, ignoring the penalties for leaving elements at the end of the last sequence without any partners. To do that, we can start from the highest score on the last column. Note that the global alignment would give us a score of -1 here, because it's considering everything, including this penalty for leaving T without a partner. But if we want to ignore this penalty here, we can decide to score only up to some point on this sequence here on the left. And the point is determined by the best score possible. So we're going to consider this only up to C. We're going to ignore this part, and we're going to consider only this part here. So this is basically what we're doing. We are scoring only this part, because using these zeros at the first row, we are ignoring any penalties that may occur for leaving parts of the beginning of this top sequence without partner. And by taking the best score of the last column, we are ignoring penalties for leaving parts at the end of the left sequence without a partner. That is by adding gaps there after the top sequence. So this is if you want to ignore gaps here at the beginning of this sequence and at the end of this sequence, you could do this. You could do the reverse. You can start with having zeros here, which means that you are ignoring gaps at the beginning of the top sequence, or you are ignoring, leaving out these first elements of the left sequence without a partner, because with these zeros here, you can start by matching this C anywhere along this sequence without penalties. So we don't need to put the C there at the beginning and pay penalties for these gaps. We can just put the C here where it matches the other C and ignore any penalties from these gaps because of these zeros. And now we can also ignore penalties at the end of this sequence by not counting what happens when we leave out elements of the top sequence. So if instead of going all the way to the end of the matrix, we stop at the best score on this last row, we are basically ignoring what the problem of leaving out these two A's without a partner. And this allows us to focus on this part. So this is a semi-global alignment where we are ignoring the need to put gaps at the beginning of this sequence and at the end of this sequence. We can also do it different combinations. For example, if we use if we put zeros on this first row here, we are ignoring the cost of leaving elements at the beginning of this sequence without partners. That is, ignoring the cost of adding gaps before we start aligning this sequence here. And if we take the best element of the last row, we are ignoring the cost of leaving elements of the top sequence without partner. That is, we are ignoring the cost of these gaps here. So this is one way which we can in which we can use global alignment to find where a smaller sequence best matches a large sequence, ignoring the penalties for what is left out, since we cannot cover the whole sequence, the larger sequence, with the smallest one. So these are, it's the same algorithm. The only change we need to do is setting the first row or the first column at 0, depending on what we want. And then instead of starting to trace back from the end, we start either from the best score on the last row or the best score on the last column, depending on which sequence we want to ignore the trailing elements. So using these combinations, you can ignore these parts and adapt the alignment algorithm to specific needs, like matching the end part of one sequence with the beginning of the other, or finding where a small sequence best fits inside the larger sequence, and so on. Okay, this is for global alignment. To sum up, we saw this problem of using similarity as a proxy for homology. What we would really like would be to be able to find which parts of the sequence of the two sequences evolved from the same parts of the ancestor and how they are evolutionarily related. But since we don't have information on that process, what we can do is to look at how the sequences are now and try to match them by similarity. When we look at substitution matrices and so on, we'll see how we can also try to include that in our similarity measures and make this approximation. But remember that what we can put on the computer for aligning sequences is based on the similarity between the two sequences and not, as it ideally should, on the evolutionary relation between them. So sequence alignment is to maximizing this similarity as measured by some rules, by some scoring that we have. In our simplified examples, we just added 1 when the residues were identical, subtracted 1 when they were different, or subtracted 2 when we inserted a gap. But there are different scoring schemes. We're going to see this next week. And we do a global alignment when we score over all the alignment. We can do a semi-global alignment when we ignore some parts on the extremity. So we can leave some things out at the beginning or end of each sequence without penalization. From the algorithmic perspective, this is a good example where dynamic programming allows us to solve the problem efficiently. Note that looking at all possible alignments would be a huge search space that we cannot possibly do in practical time with even the smallest realistic sequences. But with this kind of alignment, we can align even large sequences because it's basically, it takes a matrix with which the total size is just a product of the sizes of the two sequences, and most computers will handle the usual sequences that we need to align pretty well. So if you want, read the sections 2.6 and 2.4 of Introduction to Computational Biology, the textbook. You can read the whole chapter. We're going to cover the other parts in the next lectures anyway. So if you want to go a bit ahead, you can read the whole chapter.